For this experiment, we're going to be calculating LC50, or the lethal concentration at 50% of ammonium sulfate. I would like for you to look at this bottle of ammonium sulfate and read the bottle, see what it means. And here in a minute, I would like for you to pause the video and look at the CAS number, also called the CAS number, right here. This is where you should pause your video. What does this number tell you about the chemical? What kind of information can you find? So then here you can see these are the Daphnia that we ordered to actually test the ammonium sulfate on here in the lab. And then here I set up all of your solutions. You're going to have your 0%, which is going to be your control, and then all the other solutions, which you'll then put the Daphnia in to calculate your LC50 over a 24-hour period. Underneath the dissecting scope, you can see that I have a transfer pipette here filled with Daphnia. Now with this, you have to make sure that you actually expel your culture fluid so that you're not diluting down your sample. But I wanted you to actually see the Daphnia in the transfer pipette and then going into a solution. As you can see, they kind of swim right out. These are really interesting organisms to work with. They're really easy to see. You can see them with the naked eye. And here you can kind of see them swimming around. So it's really easy to see whenever they have ceased moving. So it's really easy to calculate the mortality rate. You don't need protoslow or anything like that. You can usually just kind of observe with the naked eye. I am now going to be putting Daphnia into the 2% ammonium sulfate solution. In order to try to not dilute down our sample, you're going to try to squeeze out as much of the culture fluid as you can. Then, because the Daphnia are crustaceans, you're going to make sure that you insert the tip of the pipette into the water and gently expel the Daphnia. The purpose of this is so we're not actually introducing any kind of oxygen or anything underneath their carapace. So we don't want to actually incite any kind of death except for what's coming from the ammonium sulfate solution. Now to repeat that, going forward with every subsequent solution. So now I'm moving into the 0.03% ammonium sulfate solution. So I'm collecting my Daphnia very gently, expelling some of the culture fluid, and then gently inserting the tip into the water, and out go my Daphnia. .4% ammonium sulfate, same procedure, inserting the tip of the cutoff pipette into the water and gently, make sure a little bit of that air comes out, expelling the Daphnia. They're coming. So one of the things I want you to observe, these are the Daphnia in our control. So control means there's going to be 0% ammonium sulfate in the solution. You can see they're moving around, lots of movement there. Okay, so when you're trying to calculate the LC50, you're looking at Daphnia without movement. Make sure that you're making observations. For example, this guy right here may not have been alive when I put him in, so it's important to take those considerations and write down the exact amount of Daphnia that you have alive that are in your solution. So these are the Daphnia that I've now inserted into the 0.05%. They're moving around right now, but pretty soon their movements will cease to exist. Typically you'll observe over a 24-hour period, and that's when you will see half. At what percent do half of these guys die?